Hey guys, as a quick introduction to what you're about to watch and or listen to, this is the second part of what I recorded with Alec Chaves. Alec is a PhD student, or candidate I should say, in bioenergetics, so metabolism essentially. So this is the second part. We essentially spend 45 to an hour dissecting a particular paper looking at caffeine's impact on muscle growth. So if you want to chime in and check out the paper with us as we go figure by figure, then I will have that linked on the podcast as well as on YouTube. Okay, without further ado, and if you haven't checked out the first part, that was on the ketogenic diet and lifting performance, so check that out as well. Okay, let's jump into it.
With that said, let's jump into how caffeine has an impact on muscle growth. This is an involved paper, for sure. This is definitely one of those papers that, well, I've got a lot of minor issues with it, but it does have some strengths that I really appreciate. So the name of the paper, and if you'd like to access it on Instagram, you can check it out. It's in my description box. You can go to the mentioned reference something. You'll figure it out. It's the green button. And on YouTube, I've got it linked here in the chat, so you guys can go through and go through the paper with us, as well as I've got a guide there to help figure out each of the figures, or read each one of the figures. So the paper is called The Effect of Caffeine on Skeletal Muscle Anabolic Signaling and Hypertrophy. So they essentially wanted to find out what happens when you consume caffeine and its impact on muscle growth. Do you see increases in muscle growth? Do you see decreases in muscle growth? Their hypothesis was that you see decreases in muscle growth. So we will find that out with them. So with that said, let's jump into the methods a little bit. The methods, I'm going to rattle off a few things. You jump in if you have anything you want to add.
Okay.
They looked at C2C12 cells. What are C2C12 cells? You want to mention that real quick?
Yes, so C2C12s, they're a standard cell line that's used for a lot of muscle biology research. They're technically derived from mice muscle cells that have become immortalized, and what that means is that they've pretty much continue to replicate and divide until at which point they're differentiated to fuse and become full, similar to like muscle fibers. And those are what you typically use to analyze and use as a model to look at skeletal muscle in an in vitro or petri dish setting.
Yeah, exactly. So they're essentially, in really simple terms, muscle cells. It's very particular muscle cells. I actually use them in my undergrad stuff, doing some microscopy work. Then the next thing that they looked at were mice. So actually they looked at rodents in general. So they looked at mice, and then they also looked at rats. So they've got 2 different models for that and 3 models in total. So cells, mice, rats. And specifically they had, for the mice, they had wild type mice, which are considered essentially normal mice. They have no alterations to them. And then they have what's called an LKB1 knockout mice. So it's a muscle specific. Okay, let me deconstruct that a little bit, because knockout does not mean Mike Tyson. We're talking mice that have a particular gene or molecule eliminated from it. So that, and it's specific to a tissue. This tissue, it could be kidney, it could be the brain, it could be anything. Sometimes when you knock out a molecule, it leads to catastrophic results, and that mouse can't even be born. This is a muscle specific, so it's a knockout of this molecule, LKB1, which I will go into once we get to that figure in terms of what it does. But they looked at this knockout model for these particular mice. And then they used electrical stimulation to induce contractions in mice. They did not do that for rats, which actually in the guide, I think I've got that wrong. For rats, they they cut the tendon of particular muscles that help support another muscle in the leg. And what that does is it allows that particular muscle, because it's not being assisted anymore by the other muscles, it allows it to grow because it has to do all the work. It goes from doing 20% of the work or 40% of the work to literally all the work completely. So that's how they're able to determine hypertrophy or cause hypertrophy through two different methods, which is cool. The fact that they used electrical stimulation, which is actually something I guess you've got. I guess you don't have experience in that directly, but Sung Kyung did some electrical stimulation, didn't he?
Yeah, so we do. We still do electrical stimulation.
Bring the mic closer.
Sorry, yeah. So we still do electrical stimulation in our cell work. So this is specific to tissue, actually putting muscles on force transducers and pretty much watching them, twitch and testing the different parameters of like maximal force, tetanic force, and things like that. But with our stuff, we use it in cells, and we more do it as a model of like in vitro exercise. So if we start getting these muscle cells to contract, what adaptations do they make? And that's really our main outcome measures. Whereas some of the mouse people who actually use these or use electrical stimulation actually want to see how the muscle performs when you give it that stimulus. So, if they're given a certain frequency, how much force can they produce? How fast can they produce it? How long can they produce before they fatigue, and things like that.
Okay, yeah, that's a really good explanation. And they use this electrical stimulation for 6 sets of 10. So think of, if you really want to make it equivalent, although it's really not, 6 sets like you would in the gym, 6 sets of 10 repetitions. So 10 stimulations, and they had 1 minute rest between each set of 10 stimulations. They removed a number of different muscles. I'm not going to go into each one. They're all in the leg of the rodent, the tibialis anterior, EDL, gastroc, things of that nature. So all of those are going to play a factor in kind of the last few figures, so we're not going to go into that too much. Anything you want to add that I've missed?
So just to distinguish the electrical stimulation model and the synergist like ablation model a little bit more. So the stimulation is an acute bout. So it's pretty much looking at what effect does caffeine have on your response to an acute bout of exercise, to an acute resistance training bout. Whereas the synergist, in which you're pretty much ablating a specific muscle to overload another muscle, that's done over a longer period of time. So it's more looking at the effect of a chronic training routine, or what effect caffeine has on a chronic training effect, versus the electrostimulation, which is more of an acute effect.
Yeah, that's actually a really important distinction, so thanks for bringing that up. Okay, so let's jump into the results. If you're following along with us, you may not be whatsoever, but if you are, then we're going to go into figure 1. So we're going to try and explain some of the basic stuff of what you're looking at. We're not going to go like a bar graph or like a little bar by bar, but just kind of overall insight. So figure 1 is in C2, C12 cells. So those are muscle cells. And you are literally looking at cells in a dish. You're not looking at cells on the actual muscle itself. So these are detached, if you want to put it that way, and they're just on a dish. And then they're measuring the quantity of a particular molecule. So they're looking at, let's see here, 5 different molecules. And 2 of them are really important, and I've got them in the guide. I've got all 5 of them in the guide, what they do. But the first 2, AMPK and AKT. AMPK, this paper is essentially looking at muscle promoting molecules and muscle inhibiting or muscle growth inhibiting molecules. AMPK, and jump in if you have a different interpretation of this, but AMPK would be considered in this context a muscle inhibiting molecule, and mTOR would be considered a muscle promoting molecule.
Kind of yes and no.
Okay.
It's more of the context you're looking at. So AMPK is kind of, you need both in order to have optimal muscle health. AMPK stimulates a protein, and its activity is stimulated in low energy states. So during exercise or during something like caloric restriction or starvation, AMPK is highly, highly active. And what it helps to do is it helps to promote things like autophagy, or pretty much cleaning up dead debris in the cell to making sure it's running optimally, and also increase our ability to both take in and also metabolize nutrients, which is an important distinction. But what it does is, because when we're in a low energy state, we don't want to be growing, is it inhibits a lot of the things that help cells proliferate, divide, and hypertrophy and grow. So a lot of the times AMPK is considered like an antagonist to muscle growth, but really it's just context based. So you can't really be growing when AMPK is active, because the overall state of the cell doesn't promote growth or shouldn't promote growth. Whereas mTOR, on the other hand, is active when you give people things like amino acids or insulin or high energy states. So it's active during those periods of times, and based on that context, you want the cell to grow. You want it to be able to hypertrophy. So again, it's just a matter of mixing the two and making sure you get an optimal balance of both. Because you need like that cleanup and autophagy phase of the cell, or of a time, but you also need the time to grow and divide and replicate, so that you're not just withering away.
Yeah, that's extremely well put, because and that's a big reason why having somebody in bioenergetics is a great person to have on this podcast, because then you get some of those nuances, because again, context really matters. AKT, do you want to touch on that? I wrote down that AKT essentially inhibits AMPK in this situation, and that it promotes mTOR. But of course, again, that's incredibly simplistic.
So in terms of where, I'm not entirely sure where AKT can feed into AMPK or inhibiting?
It doesn't. So it acts on another molecule, which then acts on AMPK or inhibits AMPK.
Okay,
so it's not a direct effect as far as I understand.
But pretty much what AKT is, it's a weird name. So actually, the true name is protein kinase B, but that's another point. So AKT is classically known as being part of the insulin signaling cascade. So when your body releases insulin, after a meal, for example, insulin binds to receptors on various tissues, and it stimulates a series of events in the cell. And one of these events is to trigger the activation or phosphorylation of AKT. And what that does is promotes a series of downstream effects to overall promote the cell to grow and divide. So so AKT is known as a mitogen. It means it's supposed to promote growth. So it's supposed to promote cell division and growth. That's essentially what I'm trying to get at there.
Yeah,
within so many words.
That's all good. Okay, so now we've got a little bit of background on those two, and I'm just going to summarize the other three. They are down. mTOR, if you think of that as a muscle promoting molecule, a master molecule, that's what it's often referred to as. These three molecules, S6K, S6 kinase, S6, and 4 eukaryotic binding protein 4-EBP1, those are downstream molecules of mTOR. So mTOR is going to have an impact on those, and then their activation or deactivation then has an impact on our genetic code, but more specifically on the ribosomes that essentially attach to RNA and then translate that to proteins or polypeptide chains and so on and so forth. We'll just say proteins though, for simplicity's sake. So let's jump into this. So figure 1A, what they're doing is on the x-axis, the bottom line where all the graphs are coming from, they're showing different concentrations of caffeine. So they've applied caffeine to each one of these cells, to each one of these cell like plates, wells, and as well as insulin. So the insulin is depicted by the dark gray or the gray bars, and the no insulin, of course, and just the caffeine is depicted by the white bars. And then they show the quantity of AMPK, which Alec just went over. So Do you want to say anything about that? Like, do you think that caffeine has an effect here?
It doesn't seem to. Based on the statistics, it does not seem to. And it's tough, again, because caffeine's effect on AMPK is so indirect, based on the upstream thing that it's activating. I wouldn't expect this to happen. I wouldn't expect for caffeine to have an effect on phospho AMPK, and I think the story from caffeine to AMPK to mTOR and growth signaling is slightly disconnected, and I think this graph kind of shows that if caffeine is going to be having any effect, it's not going to be through AMPK.
Yes, right. But then do you make anything of, so there at 0.075 millimolar of caffeine addition, they see, how would you interpret that? They see a p-value of 0.13.
So that's a trend towards significance. It's
with
not
insulin, not with caffeine in this situation.
Yeah. So what they're showing is that the caffeine that you add is able to overcome the blunting effect of insulin and phospho-AMPK, and it's able to potentially promote an increase in phospho-AMPK, which really is just a surrogate measure for activation. It's just showing that when the phosphorylation event that happens on this molecule, when it goes up, that means more of this molecule is active and ready to do its thing. So what this is showing is that there's that caffeine, if given alongside insulin, is able to overcome the inhibitory effect insulin has on AMPK.
Yeah, right. But oddly enough, and that effect seems to start at 0.075 millimolar.
Yeah.
I guess it continues on from there, but they don't. They just show that there's a trend towards significance in the one, and I wouldn't even necessarily consider that. Like, I don't know, it's 0.13. Like, when I see that, I'm like, yeah, that's just not good enough.
In terms of when we're looking at these Western blots, these very semi-quantitative measures, I didn't give that a second glance. I saw that there's no significance there.
Yeah.
If caffeine's having any effect, it's not through AMPK.
Yeah. So then B, in figure 1B, they're looking at AKT levels, so the amount of the molecule AKT, and Alec explained that as well. And really, I want to simplify this as much as I possibly can, even though there's some nuance there that we're definitely missing, because it really depends on where and in what context you're talking about any of these molecules, like Alec explained. Like in exercise, AMPK has a profound impact that you absolutely want to have. AKT, the same thing with glucose regulation, insulin, blah, blah, blah. But in this situation, just think of insulin as, or think of AKT rather, having an impact at kind of more muscle promotion, growth, splitting more of the muscle, as opposed to AMPK, if that's more active or there's more of it, then you see decreases in muscle. It's definitely not that simple, but yeah, that's just where I'm going to leave it for that. So AKT, here you see that with the addition of caffeine at varying concentrations, and these are physiological, from what I remember, these are physiological concentrations of caffeine, no effect. I mean, it does not seem to have any sort of impact whatsoever, but what you do find is that with the addition of insulin, and this goes exactly to what Alec was talking about, AKT having a profound upregulation with the introduction of insulin, that's exactly what you end up finding. You see far, far more AKT increased with the addition of insulin. Anything you want to add there?
No, I think that's pretty much it, is that caffeine, the big thing here is when caffeine's given alongside the insulin, it has zero effect on the insulin signaling cascade.
Just like turn your chair just a little bit. Yeah, there you go, you're good.
All right, sorry.
No, you're good.
I just,
I know.
Sorry.
I know.
So the big thing is that when...
You're good.
So that when you give caffeine, it seems to have no effect on the insulin signaling cascade. It does not, so it doesn't seem to interfere with this insulin or this growth signaling cascade when given alongside insulin.
Yeah, exactly. And then I guess I'll lump these three together because they're so closely related. But with caffeine addition, you don't see any effects on C, D, or E. With varying levels of caffeine, it doesn't seem to have any sort of impact, but insulin, again, does seem to have an impact in terms of increasing the amount of phosphorylated S6K and S6 and 4-EBP1. Well, 4-EBP1 not as much, but definitely for S6 and S6K. So that indicates kind of downstream of mTOR that you're starting to see increases in this kind of muscle protein synthetic pathway. But interestingly enough, you don't see that with 4-EBP1. You kind of see some divergent results there.
Yeah. You do, but I think the blood quality has a lot to do with that, because pretty much every one of their mothers can show that goes up with when you give someone insulin or when you activate mTOR.
Yeah.
So I don't know if that's it, but again, the biggest thing being is that you don't see a difference with caffeine.
Yeah.
So really, that one is questionable, but it's more this, again, pointing out to caffeine is having no effect on changing the trend that you're going to see when you throw on insulin.
Yeah, I think that's a good summary. There's not much there. I mean, when it comes to caffeine, in pretty much every figure, caffeine seems to have no impact on any of these molecules. But again, these are just in cells, and we're still going to continue with cells in figure 2. So figure 2, here they're looking at, again, different caffeine concentrations. For the first figure, figure 2A, they're looking at physiological caffeine amounts. So I mentioned in the guide that that's essentially a caffeine concentration that would be normally taken in. Like you could take that and you would find that in the bloodstream. But figure 2B looks at non-physiological. So with physiological caffeine concentrations, you essentially see no changes in the measure of protein, so myosin specifically, right?
Yeah, so this is, if we're going through these figures here, it's all about, so the first one is myosin heavy chain. So it's looking at, so myosin heavy chain is the muscle's contractile protein. So it pretty much is it's the motor that allows us to actually contract and shorten our muscle. Now, it only appears when you start out with these myoblasts that are essentially proliferating, growing muscle cells, right, that start as satellite cells. And when they're ready to then fuse and become full muscle fibers, that's when they start expressing this contractile protein, because what they're essentially saying is, I'm here to stay. I'm here to stay, and now I can contribute to the force that the muscle wants to produce.
Right.
So you're going to see myosin heavy chain come out, and they talk about it here. So when you're growing up muscle cells, you have to differentiate. You have to allow them to fuse for a period of about 7 days to allow them to start producing this myosin heavy chain. And what they're showing here pretty much is that with these physiological doses is that caffeine or incubating cells with caffeine has no effect on their ability to differentiate into full muscle fibers. And then in figure B, as you start going more into the supraphysiological range, that's when you start seeing that effect happen. So that's when you start seeing caffeine being able to inhibit or block or reduce the amount of these muscle cells that can differentiate into full fibers. And just to kind of bring up a nice distinction here, so there's papers all about muscle hypertrophy. And I think it's important to touch on maybe some of the mechanisms through which muscles grow. So obviously, when we were talking about like mTOR and things, like that's actually talking about the increasing the amount of protein that a single muscle fiber has. But actually, when we go through exercise training programs, what our muscles have is their own stem cell pool that can migrate and attach onto existing muscle fibers to actually contribute to their overall size. So that's another mechanism of hypertrophy. And by looking at myoblasts, you're pretty much doing exactly that. You're looking at cells that start out as satellite cells, became myoblasts, proliferated, and then fused, which they would typically do on a normal muscle cell. to then contribute to the overall size and growth and force production of that muscle. And what this is showing is that caffeine is having zero effect, or physiological concentrations of caffeine is having zero effect on the myoblast's ability to do that.
Right. And if you, so then looking at 2B, you're essentially doing the exact same thing, but you are looking at non-physiological, so super physiological. In this case, you're not looking at incredibly low levels, you're looking at unbelievably high levels of caffeine, which does seem to have an impact once you get up to 1.25 or 2.5 millimolar. Then that does seem to decrease myotubes, then differentiating into myocytes from that point on. C is slightly different because here they're again looking at non-physiological levels, but they're looking at the actual, not the differentiation, but the mitosis, the proliferation,
Yep.
the actual splitting of the cells to create more cells, kind of in a manner of speaking. So here they find that they're using a variety of different concentrations, physiological for the first 4, and then a super physiological for the last one, and well, unsurprisingly, they find that the super physiological one is the one that decreases this proliferation.
Yep.
Okay, well, let's move on to figure 3 then. So figure 3, we're starting to actually move away from cells. We're actually looking at our first rodent models. And here, this was complicated to explain in the guide, but here essentially they're looking at mice, and here I'm going to discuss what LKB1 does. It activates AMPK.
Nice.
Okay, so I'm just going to leave it at that. LKB1 activates AMPK, which we talked about earlier. AMPK in this context, incredibly simplified, would then reduce muscle protein synthesis, or would reduce muscle promotion. So here they used a knockout of LKB1. So they're essentially getting rid of that molecule, so now it's no longer activating AMPK, and then because you don't have the activation of AMPK through that one pathway, because it could be activated by other things, through that one pathway, then that means that you're going to see less of an inhibition of muscle growth. Kind of, that's the general gist of what's going on. So here they injected either saline as a control, or they injected caffeine. And then, on top of that, they either let the mice rest, the muscles of the mice rest, or they electrically stimulated them, which, if you go back to the methods section where we talked about that, Alec had a really good explanation of what that meant. So again, the 6 sets of 10 repetitions of stimulation of that musculature. So looking at this, did you see anything that caught your eye for A, for figure 3A?
No, other than showing that they did exactly what they said they were going to do.
Right.
Which is, so they didn't actually provide a blot showing that they knocked out LKB1. Right. But what they did provide is a good way of saying, all right, well, we knocked out LKB1. How do we know? Because in either case, the knockout did not see any increase in phospho-AMPK, despite the fact that they were given caffeine and electrical stimulation, which are caffeine not so much, but stimulation should be a potent stimulus for inducing phosphorylation of AMPK. And the fact that they showed no difference or no increase indicates that there was no upstream signal for AMPK to become phosphorylated, indicating that LKB1 was sufficiently knocked out.
Yeah, right. Yeah, and then the other thing, if you look at the wild type, the WT 0 hour, so the reason why they've got 0 hour and 8 hour, I guess I should go over that too. 0 hour is they stimulated and then they immediately took out the muscles from the mice. 8 hour is where they waited 8 hours and then removed the muscles afterwards. And what you find is with the wild type, which is the mice that have no alterations. They're just completely normal. They're just being injected with caffeine or saline, and then they're being stimulated or not stimulated. You see that with stimulation, you see an increase in AMPK levels, which goes to kind of that nuance that Alec was talking about, that with exercise, AMPK plays an important role. So you see this increase with stimulation, but not necessarily a significant increase between the two conditions with caffeine, mainly because the error bars are really large.
Yes.
That's probably why. And then in terms of the knockouts, you see, as you mentioned, you essentially see a knockout. So of course you don't see AMPK levels increase because, well, why would they? And then with the wild type after 8 hours, you don't see AMPK elevated, at least relative to the other conditions. And then again, you see the exact same thing 8 hours later with the knockout. In that condition, then there's just no increase in AMPK either. So really the only increase in AMPK is with stimulation if you have LKB1.
Yep.
That's the conclusion we can make from this. Then B, do you want to jump on that one? Well, actually, you say something. I've got something to say about this one. This particular graph made me angry.
Alrighty, and you won't. Would they like you when you're angry?
No.
Okay.
They will not.
All right. So this is looking at. So first one. So A was talking about phospho AMPK. Next one is looking at phospho AKT. Now. The interesting thing with this.
Can you look at the mic again. Just like hold up the paper. There you go.
You'll have to excuse my friend. He's a little slow. All right. So looking at phospho AKT in the exact same context as you looked at phospho AMPK. So wild type, knockout, and then 8 hour for both the wild type and knockout, given either rest, stimulation, or caffeine concomitant with rest or stimulation. Now, because phospho AKT is an insulin sensitive protein, they did not use insulin in any way, shape, or form, so we have no idea if any of these treatments are influencing the insulin signaling cascade or the insulin responsiveness of phospho AKT. But what you're seeing here, though, just kind of going through the asterisks and where they're finding significance, is apparently is that with stimulation you're seeing an increase in phospho AKT, and the same thing with caffeine plus stimulation, and you pretty much see that all throughout. So that when you look at the wild type, the knockout, and then following 8 hours, you're able to see an increase in phospho AKT with specifically the stimulation or the caffeine plus stimulation.
Yeah, that's really well put. And then I had a massive issue with their wild type at 0 hour. I'd like to know how that would be considered significant relative to the saline at rest when they are I don't know how 2 bars can be any more identical with one another, but in this situation they absolutely are. But hey, just throw some asterisks on there and we'll
be good to go.
Yeah, we are good to go. I was absolutely shocked when I saw that. I wouldn't believe that if you paid me. Well, maybe if you paid me like $10 million, then maybe. But yeah, I didn't believe that for a second.
Yeah, I really honestly don't know. And the other thing, I'm assuming it's the same site, the same specific part of AKT that's being phosphorylated. They don't mention it in here.
You just hold it, just literally just hold it.
All right, sorry everyone.
You're good.
So it would have been nice if they actually told us what phosphorylation site, because that seems to matter. And the other thing too is just what's happening to what happens with insulin. And the reason I bring that up is because, and this is I'll go off on a little tangent here, so I apologize.
Go for it.
So with a lot of the stuff I do, I focus on diabetes research, insulin resistance, and one of the most potent ways or potent stimuli that we can do to our bodies that improve our insulin sensitivity is exercise. And caffeine is commonly used as like an exercise mimetic or an exercise pill, mainly because some of its effects on the cell are similar to what happens with exercise.
Yeah.
So I would have loved to see here them show something that said, all right, you know, we gave these people stimulation, we gave them caffeine plus stimulation. 8 hours later, we treated them with insulin, and what was the effect? Did we see any potential increase in phospho-AKT? Did they see like a potentiating effect that caffeine or caffeine plus stimulation had on phospho-AKT when they were given insulin in the hours following? I think that would have been interesting. Because right now, there's really no way, there's really no conclusion to draw from this, mainly because you're not giving them a growth stimulus. Like you're not giving them something that would tell phospho-AMPK to turn on.
I see what you mean, yeah.
Now, there is something to, but it does, what they're showing here is that despite that, there does seem to be an increased sensitivity that these growth signaling pathways have, despite the fact they're giving them nothing, right? Which I'm assuming because they don't talk about food intake and what they allow them to have in that 8-hour period.
Yeah.
But whatever energy is available, the insulin signaling cascade, or phospho-AKT specifically, is more sensitive to whatever the situation is. So it's more sensitive or it's more active regardless of what's going on around it, if that makes any sense. Hopefully.
Yeah, I got you. Yeah, again, another point, just like in the other paper, something I didn't consider at all.
So, and the other thing too to point out as well is that this isn't dependent at all on LKB1.
Yeah,
so you're seeing
right.
the exact same trend. Maybe actually, even a higher, that might be a little bit of a lie there, looking at it, because the knockout 8-hour is higher than the wild type 8-hour.
The overall effect is much higher, but in terms of the actual trend, or the pattern that you're seeing, it's the same.
Yeah. So what's specific
between conditions? Yeah. Okay, I'm going to skip C because it's a Western blot, and
yeah, it's just showing the exact same thing. Yep.
Interpreting that is, well, unnecessary. The next one is figure 4. So figure 4, here they're looking at protein synthesis machinery or molecules. And I've already gone over the first 3, the S6K, the 4-EBP1, and the S6. And here again, they're just going through the stimulation with the saline and caffeine injection or at rest. And then, of course, they've got that LKB1 knockout again. And then E, they've got this, the amount of formed protein through the incorporation of a molecule known as puromycin. Is that how you would interpret? That is what puromycin does. I've never used it, so I don't know.
So say that one more time again.
My understanding is that puromycin is incorporated in protein, in the formation of the actual protein, so that you can actually quantify the amount of protein that's being synthesized.
Yes, that's exactly it. Yep.
Okay. So A, you see slight increases, right, in the wild type with stimulation, but independent of caffeine. Caffeine seems to have no impact. The exact same thing, actually, that's the case for all of them. You just see greater effects with the 8-hour condition.
Yep.
You see increases in S6 kinase. And roughly the same is true of 4-EBP1, except you don't see a robust increase with the stimulation after 8 hours. It's roughly the same as you do when you collect the muscles immediately. Yeah, anything to add to either one of those two?
No. Again,
Okay.
big thing being is that caffeine's not inhibiting this at all.
Yeah.
It's not reducing what you're seeing and what's going on.
Right. So C is S6. Here we see some differences, right? We see some interesting differences. So here you're seeing the Jesus cross for the wild type.
Hallelujah!
For the wild type 0 hour, because you're seeing differences between the caffeine stimulation, so the caffeine injection plus stimulation, compared to the saline, so the control, no caffeine, and stimulation. So suddenly caffeine seems to have an impact. I mean, I'll be honest, this is like a single graph, and everything else has been kind of worked together in concert, and suddenly this single molecule is different.
Yeah, so this, you're right, because I would expect these to be in other things, but again, we're talking about Western blot, semi-quantitative, and they're not the highest quality.
Right.
But it kind of plays into what we saw previously with the phospho-A and BK blot on the last figure,
Right.
and seeing what could potentially be a like super compensatory, or like there's a word I'm thinking of, like an additive effect that caffeine's having on the exercise stimulus.
Yeah, right.
So just as a kind of going back, caffeine has a lot of different effects on the body, right?
A ton.
And one of its most potent effects is being a nervous system, a CNS activator.
Right.
It helps to increase our central drive, and specifically in this context, to the skeletal muscle.
Right.
Like, people take it for performance enhancing purposes, and also for improving work capacity. So if you're giving mice this during exercise, then there's a chance that their exercise is actually better. They're actually able to do more during this exercise than the wild type. You're giving them a supplement. Like, you're giving them performance enhancing supplement. So in my mind, if you're doing that, then the chance that their adaptations to that exercise are also going to be greater as well. And considering one of the most important parts of resistance training exercise is the hypertrophy signal that follows, I would also expect that to be increased when you give them the supplement as well. So you're pretty much improving the exercise stimulus itself by giving them caffeine. It has an additive effect now on the exercise stimulus.
Yeah, that makes sense. Again, I do question the fact that you only see it in this one molecule, and all the other protein synthetic machinery molecules you don't see that effect. Also, the error bars are pretty large. So if I actually think that's happening, I would tend to, if this is of course just personal opinion, I would say no. But you know, I can't fully say, hey, that's not the data. I mean, I'm assuming that is the data, but it's just a little wonky compared to, like, with the background of like all these other markers of protein synthesis that just aren't showing the same trend. And you are seeing differences with the knockout condition. I mean, I guess with the knockout condition of LKB1, you do see a slightly enhanced level of S6. And you do find that actually with the other protein machinery molecules as well, 40BP1 and the phospho S6 kinase. The final thing I want to touch on is E, so I'm going to skip D. E, without looking at the Western. Here we see that stimulation does increase this is an actual measure of protein production. So it's not the molecules that are leading to protein production. It's actual, like, bar everything else, do you see an increase in proteins? Do you see actual muscle growth, not necessarily muscle growth, but protein growth? Is it being formed by this? And they can measure that by the incorporation of a particular molecule that gets incorporated to the polypeptide chain. If you're a molecular biologist, you care about that stuff. And with stimulation, independent of caffeine, again, you see an increase in protein synthesis, and you do see a difference with that knockout at the 8-hour mark. And they only did the 8-hour mark in this situation. Do you have anything to add to that?
Biggest thing here, this should have gone first. So I'm a big fan, whenever I look at papers, is I really do, like, the signaling stuff is cool, it's awesome, but if it doesn't lead to any differences in functional outcomes, it doesn't matter. It really doesn't.
Said that so many times.
It really doesn't matter to me. So the puromycin, although it's on a Western blot and semi-quantitative still, it is still a very good functional readout on how much protein is being synthesized and how that changes in response to these various treatments. So clearly what it's showing is that the stimulation is far and wide having a much greater effect, or having an effect on protein accretion than anything else. Right. Independent of caffeine, stimulation alone is what's driving this effect, right? And caffeine does not seem to be inhibiting it in any way, right? So if we have that data, that's great. Then I don't really care about any of the other signaling data, because at the end of the day, all that matters is this. All that matters is what happens to functional protein synthesis following the treatments.
Yep, exactly. Could not have said it any better, honestly. I've said that so many times. If the functional outcome is you see a difference, then you can start looking into the mechanisms of why. Mm-hmm. But beyond that, like, just...
Who cares?
Yeah, who cares? Like, if you're seeing all these different changes and at the very end, nothing changes, who cares? Mm-hmm. Because you're measuring nothing. Yep. You're literally measuring a bunch of things that lead to nothing. Mm-hmm. So it just doesn't matter.
Because it's getting caught in the weeds. That's what it is.
Absolutely.
You're getting caught in the weeds when the answer's up here. The answer's been here all along. If there's no change, there's no difference, I don't care about the weeds. I care nothing at all.
Exactly.
It's only when that changes that I care about the weeds.
So now we're going to shift our focus to figure 5, which is our last figure, but we have one more table afterwards, which is actually going to be pretty important. So figure 5, they're looking at, this is rats. So this is what I was talking about earlier, that they've cut the tendon of particular muscles to stop the rat from being helped by those muscles. So there's a series of muscles that are going to essentially hold you upright or whatever it might be, and you don't have just one muscle. So if you cut the tendon of the other muscles, then that leads to that single muscle that's left over to have to take all of the load, and suddenly that increases its hypertrophic drive, its muscle growth drive dramatically. So in this situation, and of course they used water and caffeine in this situation as well. So it's not a stimulation, but it's a different form, which is nice to see. It's cool to see that they added this this different form of hypertrophy, which Alec went over in the methods section, the distinction between the electrical stimulation and this method for figuring out hypertrophy. So with the plantaris, when they mention plantaris, they're talking about that particular muscle that they left alone to allow to grow over time. And the muscle hypertrophy meaning muscle growth, zero difference. With caffeine, no difference whatsoever.
Shocking.
Yeah, it increased. It grew 15%, which is quite remarkable. That's awesome.
Yep.
But with the addition of caffeine, it did absolutely nothing. Yeah, not shocking at all because of all the data that we've been looking at so far. Yep. B looks at the muscle mass. Here you see a slight increase, but that's compared to sham. So they've got the overload, which is in the dark gray, and then they've got the white bar, which is showing a sham surgery. So this is actually something I did for my master's. We did a sham surgery where you do the exact same surgery on the other leg, but you don't cut the tendons. So essentially the conditions are the exact same. It's a really beautiful model, says the biased researcher. But here you see that, of course, with overload you're going to see growth because it doesn't have the assistance of the other muscles, and therefore you're going to see increases in its weight. I mean, it doesn't take a rocket scientist to figure that one out. C is a little bit more interesting. I'd like to definitely get your opinion on this one. AMPK. So here you see no differences with the overload condition where they cut the tendons of the musculature. But for some reason with caffeine, and still non-significant, but do you see that decrease in the caffeine condition in terms of AMPK phosphorylation?
Yeah, I mean, I do, but I don't know if I really.
You're good.
Okay. Yeah, it's not technically a decrease. I see what you're saying, but it's not technically.
No, it's not, because the error bars on the other one, the sham, are pretty large. So I understand how it's not, but it's a stark difference.
So the only thing I could say, and this is a stretch on this one.
Okay.
So they've done several studies on chronic, and was this just a typical? What was the caffeine dose on this one?
It was physiological.
So it was still within that physiological range. So they've done stuff like this before, in which they give caffeine chronically to see what happens. And like I mentioned, caffeine's like an exercise mimetic in ways.
Yeah.
So what it does is it helps to trigger a lot of the same adaptations that you make to exercise. So technically, at rest now, if you're looking at these animals, their energy state is going to be much more adapted in the caffeine condition, because you've pretty much put them in this exercise state on top of the overload this entire time.
Yeah.
So I would expect that they'd be much better off, or much more adapted to rest and being able to keep phospho-AMPK lower, or they're much more adapted to that energy state now, compared to just the water control. There's a better way to describe it, but there's...
So we can just say that's non-significant.
Yeah, we'll just go with non-significant.
We'll just go with that. We're rounding out on 2 hours,
Yeah.
so we gotta close this out soon.
Yeah.
AKT, there's a difference with just the water condition. So with overload, you see an increase in AKT activation, but you don't see that with caffeine. Have any thoughts on that, why that might be? I couldn't honestly come up with anything.
No.
It's just a scientific mystery.
Yeah. Considering, again, this is the big difference being a chronic model, so not an acute. So
Yeah.
like you're just looking at these guys at rest, essentially, or
Yeah.
what their new rest is.
Yeah.
So no, I don't.
No.
No.
Okay, and then 5E, you see 4-EBP1, good old
Go up.
4-EBP1. Of course, that's going to increase with
Yep.
hypertrophy, so that's not too shocking.
Yep.
Finally, table 1, which is the last thing to discuss before we jump into the conclusion. They're showing body weights, the initial body weights, then after the study, the final body weights, the body weight percentage gain, as well as the heart weight. I'm not too concerned about that. The fat amount, the food consumption is definitely important. And yeah, that's about all the things that I think are genuinely important. But there are some interesting findings that essentially skew the entire study
Yeah.
from here on out, and could be somewhat beneficial actually towards the caffeine condition. I think, because you're seeing decreases in body weight, right? You're seeing decreases in fat mass, right?
Mm-hmm.
And you are seeing decreases in food consumption.
Mm-hmm.
So, I mean, clearly caffeine, and we know this, like caffeine has not a filling effect, but it makes us less prone to hunger.
Yeah.
In general, hunger pangs in general.
Mhm.
So, and then of course caffeine has a mild increase on metabolism, so that could certainly play a factor, especially with small mice.
Yep.
And rats in general. So you see that overall caffeine seems to have an impact in that regard, but I don't see any reason why.
So just going back to that, actually, because you were talking about the signaling data.
Yeah.
In the figure, at least, and I'm just going to go back just to double check, they don't mention as to when it's taken. So if you're having mice, for example, consuming more food, right, then there's a chance that you took the wild type when they just finished a meal, and the caffeine group, or not the wild type, but the water group when they just had a meal, and the caffeine group who haven't had a meal in quite some time because their hunger's been suppressed from the caffeine. So there's that could explain why you saw an increase in the AKT, phospho AKT, in the water group, but not in the caffeine group, because maybe the water group is just eating more, so they're undergoing more of those growth stimulation periods than the caffeine group. But importantly enough, none of it really matters, because again, the muscle weight, at least for the muscle they were interested in that was overloaded, both saw a similar increase. Now,
Right.
I think this could have been a little bit more significant if they used a stimulus that was less robust, right? So we're using pretty much a shotgun-like approach
Right.
to hypertrophy,
Right.
in which when you overload a muscle, you need something that is going to be really, really, really significant to kind of resist that from happening, to stop any differences. Because I forgot, what were you seeing, a 15% increase?
Yeah, 15%.
Yeah, so you're seeing a pretty significant increase in muscle size in a very short period of time.
Right.
So if you were to use a little bit more of a modest approach to hypertrophy, maybe the effect of caffeine would have made its way out. It would have shown its head a little bit more.
Yeah, right.
But because again you're using the shotgun approach, this super robust effect, the effect of caffeine might be washed away because it's not nearly as robust as the overload that they used.
I see what you mean. Yeah.
It's pretty much like you have 2 buckets of water, right? And now
Okay.
imagine the 2 buckets of water are like you're dumping. Say you're dumping 2 buckets of water in a tub, right? In one tub, you add nothing else, but in the other tub, you add another drop of water, and that other drop of water is the caffeine. If you look at them both, they both have at the end of it the same amount of water, because the initial amount that you dumped in was so high that the little bit that you could have potentially changed it with caffeine wasn't going to show at all. It wasn't going to appear because it was just not enough to overcome such a robust stimulus that is done by the overload.
Yeah, that makes complete sense. I think overall the paper shows that caffeine most likely does not have a negative impact on hypertrophy. But it also, and they actually say this in the conclusion, they're like, it might have a positive impact. And I don't know about that. But it seems like caffeine certainly does not have near the effect that actual stimulation does. If you stimulate the muscle, exercise, if you use some sort of activation or use of that muscle, the more you use the muscle, the more it grows, essentially. So if that's through electrical stimulation in the kind of trying to mimic exercise, or if that's long-term, just continuous use with the rats cutting their tendons, then you're going to see hypertrophy. But caffeine itself is just not going to have that much of an impact.
Yes, I completely agree with that. And I think that's, and it's a big sigh of relief for people that are obviously use caffeine to fuel to help them get through workouts and things. Another big thing I just like to point out with this paper, and I think it would have really added to it, is again including the caffeine after the workout period. So after the stimulation period, giving the animals caffeine, with a growth stimulus, with a meal. What effect that has, because there are some papers to suggest that having caffeine or having coffee after workout is actually not so great. Beforehand is great, afterward not so great, because you're essentially sending it these antagonistic signals. Because caffeine's a fight or flight, like you want to get going, so you want to mobilize your energy stores and get things moving. Whereas at the end of your workout, you're consuming a meal, it's a rest and digest. You rest, you build up your glycogen, you increase muscle protein synthesis. So you're sending these antagonizing signals, and you're not getting the best out of either by doing it. The other thing I just want to point, the last thing is that I would have liked to see them talk a little bit more and distinguish, because caffeine is a widely used supplement, but the vehicle in which it's given to people matters. So most of the time when we talk about caffeine, it's in the form of coffee, right? For typical, people. The reason it's most widely consumed is because it's such a high concentration in coffee. But coffee has so many other bioactive compounds that might actually be beneficial for muscle hypertrophy. And they've done mouse studies. They've given mice, they've given them water with a certain percentage of coffee, and they've actually found that it has a beneficial effect on muscle growth and signaling. So I think that also plays into it too, is the context. So what else is the caffeine given with? Because very few people just start taking caffeine pills and caffeine powders, and that's their only source of caffeine. It comes from other things as well, and it's in other vehicles. And I think that would have been nice to talk about in this paper. Maybe not cover it, but nice to talk about and distinguish how this is different than real life situation.
Yeah, that's a really good point. That's true. I'd actually be interested in reading a few of those papers if you have them on hand, or if you can find them. I guess it's not a foreign idea for me to think that coffee has a positive impact.
Yeah.
I think we all know that in different regards, but yeah, no doubt, like the vehicle for sure matters. If that's in like a pre-workout where you kind of essentially rob a lot of that beneficial product, whatever's inside of coffee, which I'm not even familiar with all the different compounds that are in coffee, but robbing it of those compounds could certainly have an impact, because here they're just giving straight caffeine. They're not adding anything else to the situation. Yeah. Okay, well, that's it. That's what we've got for you. The Instagram feed has already died again. It's been 2 hours. Man, oh man. Yeah, quite something. There's a lot of science in here. But yeah, I think we came to some solid conclusions, no doubt about that. So with that, thanks for stopping by, for those of you that were on live, and thanks to Alec for taking the time to actually read these papers and put some real thought into them. He definitely contributed several different aspects to each paper, so I'm definitely thankful to him for making the time to talk about it. And until next time, we'll have to, I have no idea how long this is going to take to edit. This might take forever. But until next time, guys, have a good one. See ya.